Modern biology often explains cellular replication as a mechanical process governed by genetic instructions and chemical reactions. In this view, DNA provides the blueprint, proteins execute the work, and the cell divides accordingly. Yet this explanation leaves an important observation unresolved. People with similar genetics, similar ages, and even similar diets often produce cells of noticeably different quality over time. Some cellular copies remain orderly, resilient, and functional. Others accumulate subtle defects, instability, and loss of coherence. This divergence cannot be explained by genes alone. An alternative framework begins by shifting attention away from genetic instructions and toward the internal environment of the cell itself. In this framework, the cell is not a passive machine executing code. It is a self-organizing system whose behavior depends on the condition of its internal medium. Proteins, water structure, signaling fields, and energetic coherence together form what can be described as the cellular inner environment. This environment is not chemically neutral. It behaves as a condensed biological medium capable of organizing information, not merely transporting molecules. Cellular replication, in this view, is not only a chemical event but also a contextual one. The quality of the copy depends on the quality of the environment in which copying occurs. From this perspective, the role of plant-derived substances requires reconsideration. Herbal extracts are typically analyzed as collections of isolated compounds. However, such analysis fails to capture a critical property. These substances are formed through prolonged interaction with light, soil, water, time, and biological rhythms. They are not merely chemical mixtures but structured biological condensates. They cannot be fully reproduced through synthetic reactions derived from industrial feedstocks. Their value lies not in rarity of molecules, but in the organization of their internal structure. When introduced into the body, these extracts do not function as commands imposed on cells. They function as environmental modifiers. They alter the conditions under which cellular processes take place. In this framework, herbal substances act as dielectric organizers. They stabilize the internal cellular medium, reduce informational noise, and support coherent protein folding and signaling. Rather than forcing activity, they create conditions that allow cells to regulate themselves more intelligently. This distinction is critical. Cells operating in a noisy or destabilized environment prioritize survival. Replication proceeds under stress, increasing the likelihood of imperfect copies. Cells operating in a coherent environment are not rushed. Replication occurs only when conditions support fidelity. This explains why certain lifestyles consistently produce cleaner cellular copies. Such lifestyles tend to minimize disruption to the internal cellular environment. They maintain rhythmic stability, reduce exposure to synthetic interference, and sustain contact with biologically structured inputs. The result is not moral superiority or discipline, but environmental clarity at the cellular level. In this view, health is not imposed. It emerges. A cell does not need to be instructed to behave correctly. It needs conditions that allow its intrinsic intelligence to operate. When the inner environment supports coherence, cellular replication becomes orderly by default. Thus, the question is no longer why some interventions work and others do not. The deeper question is whether the internal environment permits clean replication at all. Longevity, from this perspective, is not the accumulation of years. It is the sustained preservation of an internal environment capable of producing coherent cellular futures. And that environment, quietly and continuously, determines whether life renews itself or slowly degrades from within.